Let's get started. Good to see you guys. It's an intimate gathering. Wonderful. Well, I'm so grateful you came. I hope you guys all ace your test this afternoon. And I'm here afterwards if you want to ask me any questions about the material that you might be tested on. And so take advantage of it. Okay, we're going to talk about proprioception today and learn about how information about length and force in your muscles when you perform an action gets sent back to the nervous system and the representations that form because of it. So just a brief review about where we were with respect to a muscle. So here's extrafusal muscles, these red things that you see here. This is what generates force. They're innervated by alpha motor neurons, the motor neurons that together make a motor unit. So an alpha motor neuron innervates a number of muscle fibers. Those muscle fibers are extrafusal, which produce the force. Then there's this intrafusal fiber. Intrafusal fiber is part of the sensory system. It gets innervation by another kind of a motor neuron called the gamma motor neuron. It also contracts, but that contraction is only to set the threshold for the afferents, the muscle spindle afferents, which what gives rise to the sensory information regarding length of the extrafusal fiber. So basically, this intrafusal fiber is sitting in parallel with the extrafusal fiber. As the extrafusal fiber gets pulled and pushed, as this length gets shorter and longer, the intrafusal fiber also gets shorter and longer, and that gets sensed by the muscle spindles. So we're going to talk about the extrafusal fibers and the intrafusal fibers today. I'll describe to you what exercise does to your muscles, what spinal cord injury might do to the muscles, the atrophy that takes place, and what handedness does to the composition of the muscles. So the myosin that's in the muscles, the extrafusal fibers, has a particular configuration, and that gives rise to the type of the muscle fiber that you have. So in general, we have 2-3 different kinds of muscle fibers that make up that extrafusal fibers. There is type 1, which is the slow contracting fibers. So when a stimulation arrives on a type 1 muscle fiber, what it does is that it makes it contract but doesn't produce a whole lot of force. Type 2 also causes contraction that produces force, but these are faster. They generate forces that are earlier, and they produce forces that are greater. There's a 2A, which fatigues relatively little, and 2X, which easily is fatigued. And so type 1, what's special about it is that it doesn't fatigue. So the fiber types that make up a muscle depends on its function. So think about a muscle that you use all your life, and a muscle that is a part of the force production system of your body. It causes contraction, it causes movements, and you use it all your life and you never want it to fatigue. Which muscle would that be? What do you think? Well, the heart isn't connected to any skeletal part of the body, right? Which other muscle? What do you think? Diaphragm. So you never want to get tired of breathing, right? Your diaphragm is made up of entirely type 1 slow contracting muscle fibers, and through stimulation we'll see that it always will cause a contraction, and it never has the fatigue properties of the, say, your leg muscles or your biceps. All right, so let's look at these types of muscle fibers and how they respond to stimulation. So a muscle fiber and its motor neuron is called a motor unit. And so here's an action potential that comes down the nerve of the motor unit, and it produces a twitch in the muscle fiber. So if the muscle fiber is slow, you see that it takes it about 50 milliseconds to generate that peak force, and then it gradually declines. The other thing you see is that it produces only about 1 gram of force. Now, what's special about these types of motor units that are controlling slow muscle fibers, is that, you know, if you give them a stimulation, boom, 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 and it adds, and then it reaches some threshold, and then you can maintain that force for as long as you want. So in this case, the data is continuing for 60 minutes, and you see that the force that the muscle is producing isn't changing. The fiber can generate that force, so it doesn't fatigue. Type 2 muscle fibers come in two varieties, fatigue resistant and fatigable. So both of them are fast. And so what does that mean? You notice that from the time that the stimulation is given to the time that this rise takes place, it's quite early, and look at the magnitude. The magnitude of the force is 10 grams. So an order of magnitude larger than this. I'm sorry, we don't have a time scale here for it. The other thing is that this is fatigue resistant. So you see that it reaches some amount of force, and then it only gradually begins to decline, as you see it over the long term here. So after an hour of stimulation, it has definitely fatigue, but it's a fairly gradual, slow process. Finally, here's a fast fatigable. It has a rise that goes up to 50 grams, so 5 times the fast fatigue resistant, 50 times the slow, but now it can't sustain it very well at all. You see how it rapidly drops off. So 3 different motor neurons are stimulated intracellularly. Twitch response, this is the twitch response. This is tetanic response when it's maximally stimulated, and tetanic stimulation for over a longer period of time, every second, for in this case a minute, or say 15 minutes. Do you have any questions about that? So a single motor neuron innervates multiple fibers. That's what makes a motor unit. The motor units, that motor unit is the muscle fibers that it's innervating. They're all going to have the same property. They're all going to be slow, or they're all going to be 2A or 2X. And so they're all going to have responses like this, or like this. So slow, fast fatigue resistant, fast fatigable. So what are the properties of these motor units? Well, their axon diameter is small for the slow type. The action potentials get there late. For the fast fatigable, it's large. The tetanic tension, the maximum force that it produces when it's stimulated, it's small. For the slow type, it's large for the fast type. The speed of contraction is slow, fast. Fatigue properties, it doesn't fatigue very much. If it's a slow, it fatigues rapidly, and so forth. And particularly important is the metabolism. So the metabolism of the slow fibers is aerobic, whereas the metabolism of the fast fatigable is anaerobic. All right, any questions? Yeah?
What does the number of terminals mean?
It refers to the axon and the terminals that it makes on the muscle fiber. So the number of neuromuscular junctions. Good question. Okay, so what happens when you go and you lift weight? Well, after a while, the muscle gets bigger, right? And so what's going on there? How did the muscle get bigger? Well, your muscle fibers, you don't get new muscle fibers, because if you did, then there would have to be new neural innervation as well, right? What's happening is that the myosin is responding to this tension at the junction between the muscle and the bone, at the tendon, and that response is to make it bigger. So the individual muscle fibers are getting bigger when you lift weights. Now, we want to compare a use of a muscle over long periods of time with lack of use, and ask, how did that muscle change? So in one hand, you may never exercise, you may never use that muscle. That would be a person that has had some kind of a damage to their nervous system. So maybe a spinal cord injury. So a spinal cord injury would result in lack of use. On the other hand, you may be a world-class athlete where you use that muscle all the time. So what happens to the composition of that muscle fiber? So this is a quadriceps, which is a thigh muscle. And what this plot shows you is the composition of that muscle in a healthy individual, the average active person, and you see that it's mostly made up of slow type, and a large component of it is type 2A, fatigue resistant, and a tiny part of it is type 2X, fast fatigable. But on the extreme, you have a person with spinal cord injury. So this individual cannot activate these muscles because the spinal cord prevents the individual from reaching those motor neurons. And so what you see is that the amount of slow type muscle fibers has decreased significantly as a proportion. And what you see is a gigantic rise in the number of fast fatigable muscle fibers. On the other hand, you have an extreme endurance athlete, a person that is trained to run for tens, perhaps even 100 mile, and what you see is that you've increased the composition of the slow, non-fatigable muscle fibers. And in between, you might find yourself. All right. So lack of use makes the motor units so that the muscle fibers become fast fatigable. Use makes it so that they become fatigue resistant. They become more slow. All right. So how does one control the force that's being produced in the muscle? Well, the simple case would be to take a motor neuron and increase its firing rate. So if it's firing at 1 hertz, you're getting a little bit of force. At 10 hertz, you're going to get more force. Now, we have lots of motor neurons. And so what this shows you is as the person increases the force that they're generating, what happens to the motor neurons? What you see is that a motor neuron is becoming activated, it increases its firing rate as force increases, and then a new motor neuron is recruited. And then a new motor neuron is recruited, and a new motor neuron is recruited. So you're getting two things as you're increasing the force that you're producing. The motor neurons that were activated early, they are increasing their firing rate. And then as the need for force increases, new motor neurons join the process of generating those action potentials. So you get both recruitment of the motor units and increased activity in the motor unit that was already activated. So as more force is needed, more motor neurons are recruited, frequency of activation of motor neurons is increased. Okay, so what's the difference between the motor neurons that get recruited here versus the ones that get recruited here? What do you think? So we have these motor neurons that are going to the muscle fibers. And so you might ask, well, are these motor neurons going to the fast ones or are they going to the slow ones? And what about these ones here? Is there any regularity with these? Now what do you think? Which ones should you recruit first? Because remember, the slow type motor units, they don't produce a whole lot of force. It's the fast, fatigable ones that produce a lot of force. And so you might guess that these motor units that are getting recruited are the slow ones. These are the fast, fatigable ones. And indeed, that's the case. So how do we know that? So this experiment shows you how you can estimate the twitch properties of the motor unit. So is this a fast, fatigable motor unit or is this a slow motor unit? And what does its twitch response look like? And the way that one does it is through a process whereby every time a motor neuron gives you a spike, you try to find what is the force that was produced by the muscle fibers that that motor neuron innervated. So how do you do that? The idea is this. You generate force with a muscle. And what this electrode does, basically a needle in the muscle, what it does is that it picks up action potentials that are being generated by a single muscle fiber. And now what you want to know is that what is the force that's being generated by this muscle fiber? Well, the problem is that when you generate force, of course, you measure force from the entire muscle. And you're now recording an action potential from a single muscle fiber. So what is the contribution of that muscle fiber to the total force? Well, the way you can tell is that you align your data to every time that there's an action potential on that particular muscle fiber. So what do you measure? You're measuring force. That's tension here. And then here's what you have. You have an action potential, and every time you get an action potential, you say, align the data that I've measured to when I got this spike. So here's the spike that you got, and here's what you trigger. You say, all right, align the data to this. And so if you just had one spike, you know, here's what you would see. This is the force that you see, right? And it's very noisy. If you had 10 spikes, then 10 spikes meaning you got this muscle to produce, this muscle fiber gave you 10 spikes. So every time you got a spike, you take that force data over this window and then you average it. And so you see that, oh, you see slowly a pattern developing. 100 spikes gives you this, 500 spikes gives you that. So this is spike triggered averaging. And what it's doing is trying to identify what was the twitch that was produced by this single muscle fiber from among all of the forces that were generated by all the active muscle fibers. And so what you see is that here's this shape. Spike triggered averaging. This is a typical way by which you can understand and quantify the contribution of one voice from among a chorus. It's like if you had a microphone that that recorded the sound of a large room and you wanted the voice of one person, well, what you would do is that every time that person would say ah, they would press a key. And that ah would eventually rise from among the noise if you had sufficient data. Does that make sense? Do you have any questions about that? All right, so here's a technique then that one can use to quantify the response of a single muscle fiber. All right, so now the question is, during voluntary contraction, so here's a force that's being generated, here's a motor unit that gets activated early. Here's a second motor unit that gets activated late, so its spike looks different than this first one. And the question is, what's the twitch that this first one is generating, and what's the twitch that the second one is generating? And so the answer is shown here. So here's the first motor unit. You see that it has a small force that it generates, and it has a time to peak that's long, so the response is slow and weak. And here's the second motor unit. Its response is large, and its rise time is rapid. So it generates a force quickly and by large magnitude. So what this means is that you get those slow type motor units activated early during force production, and then later, as you need greater amount of force, you get the fast, perhaps fast fatigable motor units later. Does this make sense? So we see two things here. The firing rate increases, a new motor unit is activated, so you get recruitment of a second motor unit, and the second motor unit is faster and likely going to be of the fatigable type. This is the orderly progression of recruitment. All right, so if use alters composition of the muscle, you guys, almost all of you guys, are going to be right-handed. A few of you may be left-handed. And so what that means is that your hand, that's your dominant hand, you use it more often than your non-dominant hand. So does the composition of the muscle that makes up your dominant hand, is it different than the one that is your non-dominant hand? And this study measured that. And so how did they do it? So what they did is that they measured the threshold at which in terms of force, at which a motor unit became activated. So this is what they're plotting here. The dominant hand is the black one, and the non-dominant hand, which is the open bars. And what you see basically is that in a person, their dominant hand is made up of more units that get activated early, whereas the non-dominant hand has more, relatively speaking, units that get activated later. The task is an isometric force production in the first dorsal interosseous muscle. So the composition of the first dorsal interosseous muscle is more units that are probably slow on the dominant hand, and for the non-dominant hand, relatively speaking, the muscle has a greater proportion of those motor units that get activated later, probably the fast type. So use of the body part tends to make the muscle fibers of that body part more like a slow type of fibers. And so one of the questions that, you know, people who train for explosive kind of actions, like say short distance running, high jump, is that, you know, how do you train so that the muscles become best suited for the action that you're going to do? It's clear what you need to do to train for long distance running. You need to do long distance running. But what do, you know, so you have explosive power? And it's, you know, obviously you don't want to damage your spinal cord so that you get more of those muscle fibers. And so the training that they do is interesting. You know, they've learned that they need to train for a while, but then stop training after some amount of time, right before they go and do their meets. Somehow that transitions, basically, you don't want to get too many slow fibers. You don't want to get, you know, reductions in your abilities, and yet, you know, being able to train and do the task well. So use tends to transform the muscle fibers into the slow fibers. All right. Any questions? Yeah.
In that transformation process, are old muscle fibers converted, or do you get new ones, or like how does the
The myosin changes. Yeah.
Okay.
The proteins that make up that element changes. Yeah. So the only time you change the actual number of muscle fibers, it tends to be age.
Okay.
Because the age causes loss of muscle fibers as well as loss of motor neurons. Particularly the fast type. Yeah. So you don't find many old people wanting to sprint. Okay. So let's now talk briefly about the afferent system. So the information that needs to come to your spinal cord to tell you two kinds of information. What is the length of the muscle, and what's the force that that muscle is producing? So this diagram shows you extrafusal fibers. Here they are. Here's the alpha motor neuron that comes to them. So here's a motor unit. So this axon comes from alpha motor neuron, innervates this fiber, this, and this. So these four fibers are the motor units that make up this alpha motor neurons family. Then there is gamma motor neurons that go and innervate these intrafusal fibers, and these intrafusal fibers house muscle spindles. Basically, spindles are afferents that wrap their body inside these intrafusal fibers, and when this fiber gets stretched, this structure generates action potentials to tell the neuron in the spinal cord that the fiber got stretched. Then there is an afferent system that innervates this interface between the extrafusal fibers and the tendon, and this now gives rise to another kind of a afferent called the 1B afferent. So this is 1A and type 2 afferents called spindles. These are tendon organ afferents. And this is another input to the nervous system. And what this is telling you is what's the force that's being generated by these extrafusal fibers. So the Golgi tendon afferent, the Golgi organ afferents, are going to be telling us information about force. The spindle afferents are going to be telling us information about length. And I'll show you data for that. All right. So here's the muscle spindle afferent on the left. So what's happening is that you have a muscle that length is increasing and then held constant. And so look, here's the spikes that's being generated by the 1A afferent. It gives you lots of spikes during this rise period where velocity is high, and then velocity goes to 0. It gives you less spikes, but that's just length change that's being reflected. So there's some length that's being represented the length is higher here, larger here than here, so you see the activity is higher, and then there's this period when the length is changing. So spindle afferents are particularly sensitive to velocity, but also sensitive to length, which you see here. Now, this is called an isotonic stretch. What that means is that the alpha motor neurons are kept constant. Their activity isn't changing. All that's changing is the length of the muscle. On the right, what we have is force change. So in this case, the muscle itself the length isn't changing. It's kept isometrically. But the muscle is generating greater force. And so you stimulate the muscle, it generates greater force, and then you ask, how does this force information gets transmitted to the nervous system? And what you see is that as the force increases, kept constant, and then decreases, the Golgi tendon organ afferents, they increase their activity, maintain their activity, and then they drop off. So here there is no length change, only a force change. You see that the Golgi tendon organ afferents are providing force information. Here's length change, and here you have muscle spindles provide that information. Okay. Here's an experiment that you can do yourself to affect your afferents, in particular the spindle afferents. So the spindle afferents are particularly sensitive to vibration. And if you were to vibrate the belly of your muscle, you will feel that there is this change that's taking place in the length of the muscle. So this experiment, what they did is that they had a person sit at a table like this, and with their elbow up on the table. And their objective was that the right hand was pulling a string attached to the ceiling. So just like this. It's just pulling down on the string. And the task is to match the position of the right arm with the left arm. So if your arm is here, with your eyes closed, match it with the left arm. And you could do that easily. And now what they did is that the right biceps is vibrated, but remains stationary. So the arm is the same length. It's not changing length. They vibrate the right biceps. So if you vibrate the right biceps, by that what I mean is like 100 hertz, you could do it with your cell phone. Just put it there and you vibrate it. What happens is that causes activity on those muscle spindles. And it tells the brain that the length of this biceps is increasing. Right? So it feels like the arm is down here when it's actually here. And so during the task, if the job is to try to tell me where do you think your arm is located, the person moves their left arm down because they feel that the biceps on the right arm is increasing its length. And so you see that. Here's where the biceps is vibrated on the right side. The tracking arm, which is the left side, it extends to say, you know, it feels like that arm is increasing. So you see that here. The left arm moves down to say that I sense my right arm increasing the length of in this case, the biceps. Okay, does this make sense? Your perception of where your arm is depends on the activity of the muscle spindles. Okay, so one of the coolest, yeah, go ahead.
Why does the vibration means that the muscle increases length?
It activates the spindles, yeah, artificially. And so one of the coolest experiments you can do is to hold on to your nose and vibrate your biceps. It feels like your nose is growing. It's really bizarre. Why? Because it feels like your arm is going out. And you know, your head isn't moving, so something must be changing. And it feels like your nose is growing. Fun things you can do if you know science. All right. Any other questions? Yeah. Okay. So I've often thought one of the hardest things as a job would be these people that, you know, use the jackhammers, because that is like nothing but vibration. And I don't know how they, you know, what kind of nervous system do they have after a lifetime of jackhammering? Because that vibration, does it have any long-term effects on your muscle spindles? I don't know. Anyway, it's amazing that they can do it. Okay, so muscle spindles are innervating the interfusal fibers, and the interfusal fibers are also innervated by a gamma motor neuron. So here's a gamma motor neuron that innervates the interfusal fiber. So here's the extrafusal fiber, the cartoon, just to show you the extrafusal fiber that gets innervated by the alpha motor neuron. And then there's this gamma motor neuron that innervates the interfusal fiber. What's its job? Its job is to stiffen up the interfusal fiber, and so that it's like a threshold. If you set this to be some length, any stretch of the extrafusal fiber will now result in a rapid change in the interfusal fiber, in which case you can sense it by the muscle spindle. So basically, you don't want this interfusal fiber to go slack. So you don't want it so that at some length, so if your arm is here, you don't want the interfusal fiber to have such a long length that the spindles can't sense motion of the arm. And so what the gamma motor neurons do is that they stiffen up, they activate the interfusal fiber and set this length that says if this length changes beyond this amount, sense it and send that information to the nervous system. It's a threshold that beyond which the spindles can sense it. So muscle spindle is sensitive to both gamma motor neuron input and the length of the extrafusal muscle. All right, so here's the network. You have your extrafusal fiber, you have your interfusal fiber, you have your muscle spindles. You have a alpha motor neuron. An alpha motor neuron sends its axon and makes a contact on the extrafusal muscle fiber. That causes contraction. That gets sensed by the interfusal fiber that activates the muscle spindle. That muscle spindle is part of the afferent system that provides information through the dorsal part of the spinal cord back to the alpha motor neuron. And this is an excitatory reflex. So if you stimulate, you activate, you cause force. If this gets lengthened, if the muscle spindle gets pulled, it causes activity here that then also activates the motor neuron. So if the muscle gets pulled, this pathway will excite the motor neuron, which will then activate the muscle, which will then contract it. So it's a simple feedback system. If your muscle gets pulled, that activates the spindle activates the motor neuron activates the muscle, that contracts, and the arm comes back. So it's a simple feedback system. All right, what about the Golgi tendon organs? The Golgi tendon organs innervate the tendon. They measure force. They inhibit the motor neuron. So you have an excitatory pathway, feedback, length is fed back and excites the motor neuron, force is fed back and inhibits the motor neuron. Okay, we can put this together as a control diagram now. So we have some input that we're giving to our alpha motor neurons from the brain. We send some command to these inner neurons that are measuring force and to the gamma motor neurons that are modulating the length. So you generate some command alpha motor neurons. It activates your muscles. That muscle generates force. That force gets sensed by the tendon organs and is set back to the alpha motor neurons. That force acts on a load that causes a length change. That length is sensed by the muscle spindles. The muscle spindles are controlled by the gamma motor neurons. That information is sent back to the alpha motor neurons. So you can control length and force. And you need to be able to do both, right? So to control length would be this. To control force is to be able to push. How much should I push? You know, small amount, large amount. Any questions? All right. So now we're in position to go over a simple reflex that you get tested when you go see your physician. So, you know, you've been for your annual exam. They have you sit on a table, often examination table, with your knee dangling like this. And they take a hammer and they tap below your kneecap, called the patella. And what they're doing is that they are extending this tendon. So by tapping it, you get extension here. You pull this muscle. This stretches the muscle spindles. the muscle spindles get activated. Here's the cell body, sends action potentials to the spinal cord. It activates the motor neuron that goes to the same muscle. That activation generates action potentials on the extrafusal fibers, which then stimulate this muscle, which means this muscle produces force. It shortens, which means it lifts your leg up. So what are they measuring when they're looking at that response? Well, they're measuring the neuromuscular system that goes through your spinal cord and generates a reflex response. They're measuring your reflexes through the stretch reflex. All right. So that reflex depends on the spinal cord. But you can respond much more than just through the reflexes of the cord. You also have your brain that you can respond to. So the information that comes to the spinal cord gets conveyed to the brain and then comes back down and can be resulted in actions. And so can we dissociate the spinal response from the supraspinal response, the information that's gone to the brain and back? And this gives you an example. So the task is hold a particular location. Biceps is suddenly stretched at some time s. Before the stretch, the subject is instructed to either oppose the stretch or assist the stretch. So biceps is going to get stretched, and you know that this is going to happen. You don't know when, but you know it's going to happen, but there's going to be instruction. Oppose it, push back, or let it go, assist it. I want to show you the difference. So here is biceps EMG. So the activity in the biceps when the stimulation, when the stretch takes place. So here's when the stretch takes place, and the instructions are to oppose it. So you see that there's this, at 50 milliseconds, there's this activity in the muscle, and then later there's a secondary response. Whereas if the instructions are to assist, you see there's still this early response, but then the later response is gone. So what we label this is the involuntary response. And the secondary one is the voluntary response, because instructions could change it. So the involuntary response is largely from your spinal cord. It's going to produce your response regardless of the instructions. This voluntary response is from probably your brain. So that information has gone to the brain and has come back down, and in one case says resist, in another case it says do nothing. The important point being, notice, it takes time. And why does it take time? Because it's much longer route from the spinal cord to the brain and back down than it is to simply respond with the circuitry in the spinal cord. Let me show you that pathway and then the time. So here's what that stretch is doing. That stretch is causing the muscle spindles to send a signal to the spinal cord. And there's a direct pathway in the spinal cord that activates a motor neuron and results in contraction of the muscle. That's the short latency pathway, the rapid pathway. Then there is a copy of this that's sent via the brainstem, thalamus, the motor cortex, and then back down from the motor cortex to the motor neuron. This is the long latency pathway. So we're getting two responses, a short latency and a long latency. All right, so what are these latencies and how can we tell? So this experiment shows you some of the time components of this problem, the time delays. So what we have is that we have a subject who's sitting comfortably, the ankle is suddenly stretched. So the ankle is moved like this. Now in the top plot here, you see evoked potentials from the somatosensory cortex recorded by electrodes that are placed on the scalp, EEG electrodes. So the foot is stretched, and you notice how long does it take before you see activity that corresponds to that stretch. And you see it here. So the EEG response is flat, 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 flat. About 50 milliseconds later, you see some change. So this gives you an indication of how long it's taking for the information to go from the spindles in the foot to the spinal cord up to the brain. The second line here is the evoked potentials, which means that EMG electrodes that are placed on the ankle muscle when motor cortex of the same subject is stimulated via magnetic stimulation. So TMS, transcranial magnetic stimulation, is placed on the motor cortex, the foot region, which would be medially. You stimulate, and then you see how long does it take before the muscle, in this case the ankle muscle, is activated. So here's when the stimulation takes place in the motor cortex. Here's when the response takes place in the muscle. So about 30 milliseconds. So about 50 milliseconds here, 30 milliseconds here. Then down here, you have the muscle itself and how it responds to that stretch. So this is the EMG when the ankle is suddenly stretched. So here's when the stretch takes place. This is the time course of the stretch. You see that here's the first response, second response, and then a third response. In this case, there are 3 responses here. So if we add up roughly this delay and this delay, you get something like this amount, about 90 milliseconds or so, which says that the information, for it to go from the spinal cord to the brain and back down, this is the amount of time you need. So then you might say this response here, that this late response, after about 100 milliseconds, is probably going from the spinal cord to the brain and back. Whereas this response, and possibly this response as well, is to the spinal cord and maybe the brain stem. It doesn't go all the way to the cortex. Any questions? So you have a fast pathway, the spinal cord, and then you have a slower pathway, the brain. Okay. Let's see an example of this in a stroke individual. So this person is a stroke survivor, and they have had a stroke in the right brainstem. And what this is doing is that it's making it so that they do not have good control of their right side. So let's look at the notes that are represented here. So joint position sense, JPS, 2 arrows downward. That means there's very low, much reduced joint position sense in this patient. On the right side, on the right, let's see, what is it? The thumb flexor muscle. So on the right thumb, they have very low representation of what the position of the right thumb is. So what the clinician is doing is that he's taking the right thumb and having the person close their eyes and is moving it to a flexed position or not, and is asking the individual, is the hand flexed or not? And when you see double arrows down, that means the joint position sense is poor. You also see 2 PT, 2 arrows down. 2 PT is 2-point discrimination. So what that means is that you take 2 sharp needles and you ask how far apart do they have to be before the individual senses 2 points rather than just 1 point. Okay, so if you have excellent sensory feedback, you can have these 2 points be within a millimeter of each other and you can still feel 2 points. But if you have a dull sense of sensation, then these 2 things can be a centimeter, maybe farther apart, and you still just feel 1 point. You just feel pressure. It's just dull pressure. You don't feel 2 specific things. So these nomenclature, 2-point discrimination is down, joint position sense is also down. Okay, so this is the right thumb, this is the left thumb. And what's happening is that in this experiment, the thumb is stretched like this, and as the stretch takes place, here's time here, so this is about from here to here is about 50 to 100 milliseconds. 100 milliseconds is about out here. Here's when this stretch begins until about here, and you see that this is the EMG response on the side that's normal, the left side. Then you see that there's this first response, second response, and then a third response. And whereas for the right side, where the side that's been affected by the stroke, you see there's just 1 response. And the second and third responses are missing. So this pathway from the muscle spindle to the motor neuron to the muscle, this pathway is okay. What's happened is that a copy of that signal needs to go to the brainstem, up the thalamus to the motor cortex and back. CST means corticospinal tract. What's happening is that this pathway has been damaged. And so this information isn't reaching the motor cortex. And so you just see that first response, but you don't see the later response. Any questions about this? Okay. So normally, you can control these later responses. That's why they're called voluntary. So you can modulate how strongly or how weakly you respond to stimuli. In some cases, some pathological conditions, that ability is affected. So in Parkinson's disease, these reflexes are changed. And it's certainly unclear to me why this might be. But I want to show you an example of it in this slide. So here's a healthy subject. And here's a person that suffers from Parkinson's disease. And what's the task? The wrist is going to be extended. So the wrist is going to be extended like this. And look at the EMG that results. So in the passive condition, instructions are to not resist the stretch. In the active condition, instructions are to resist the stretch. And what you see is that here's a wrist position that's being extended in the healthy subject and in the patient. And the passive condition is here, which means to just let it go. The active condition is that as soon as you feel the stretch, respond. And what's puzzling is that if you look at the PD patient here, you see that in both cases, they're responding to the stretch. They have rigidity. This is what this rigidity would be. A change in the position of the wrist is resulting in a strong response, despite the fact that the instructions were, to let it go, to be passive about it. So the feedback is abnormally high response to that stretch of that muscle. All right. So how important is this feedback for control of voluntary movements? When you make a movement from point A to point B, how important is this sensory feedback in determining that activation in the muscles? Well, what I want to show you is that patients that have suffered a disease called large fiber neuropathy, basically in large fiber neuropathy, those muscle spindles die. And these individuals become what's called deafferented. They can't sense the position of the musculature. In particular, the disease begins in the distal parts of the body out here, and then the muscle fibers slowly begin to get deafferented. So you can ask these patients to make voluntary movements. And of course, they have learned to make these voluntary movements long before they lost the sensory feedback. And I want to show you the comparison between movements here between healthy individuals and deafferented individuals. So remember that we had, when a normal movement takes place, here's the velocity. If you look at the muscles, you should see this three-burst activity: agonist, antagonist, back to agonist. The three-burst activity to make a rapid movement. If you ask a deafferented patient to make a rapid movement, they too can make a rapid movement, and they also have reasonable EMGs. So you don't need the afferents to generate the activity in those motor neurons to make a rapid movement, at least not for the hand muscles. So what then is wrong for motor control of these individuals? You see it here. So in this case, what the healthy individual is asked to do is to generate a force and then maintain that force without visual feedback. So you generate a force and hold it there without visual feedback. It's as if you're asked to hold something and not be able to see it. Well, a healthy individual can do that. But a deafferented person, they generate that force, but as soon as the vision goes off, they're lost. They don't know the force that they're producing. And the force drifts in all directions. Okay, so you don't need the sensory system for generating the typical shape of EMG, agonist, antagonist, agonist, but without sensory feedback, you have no sense of where your body is in space. And so without visual feedback, you can't maintain that position. Any questions? Okay, good luck with your quiz today. Thank you so much for coming.